Click for IB Biology. Here we're looking at the diversity of carbohydrates, and our first molecule to look at is the hexose, the 6-carbon sugar called glucose. So we'll be looking at the structure of glucose and some of its properties. Should you wish, there's a PDF in the description that you can use to make your own annotations as we go through this, and then later you can take the quiz which is down there to check your understanding. Let's consider the diversity of carbohydrates first. So carbohydrates are compounds in which we have carbon, hydrogen, and oxygen. Generally speaking, they are in the ratio of, for every carbon there's H2 and O, more or less, and this distinguishes them from the lipids, where the amount of oxygen present is much lower. So our carbohydrates can be divided into the saccharides, the sugars, and the polysaccharides. Sugars themselves form what are called the monosaccharides, the single sugars. So if we think of monosaccharides as single sugars, and during this course we will come across 3-carbon sugars called trioses, 5-carbon sugars called pentoses, and 6-carbon sugars called hexoses. If the 2 hexoses join together, this gives us molecules that we would call as disaccharides. So these are double sugars. The most common examples that we'll be dealing with are maltose, which therefore has 12 carbons, so that's 2 times 2 glucose molecules joined together gives you maltose. But another C12 disaccharide is sucrose, but in this case it's a combination of glucose and fructose, giving it different chemical structure and different properties. Now if we join many glucose molecules, this one in here, so if we join many of these together, we get a polymer of the sugars. So the monomer is joined together to form polymers, many of glucose molecules. In plants, the classic polysaccharide is starch, with 2 forms called amylose and amylopectin. Other forms in starches are the combination of glucose molecules to form these large branching chains known as cellulose, which we have in the plant cell wall. But in animals, we have a much more branching structure in the polymer, which is the animal storage of glucose, which is known as glycogen. So that's an animal process. So let's take a look at hexose sugars glucose with the formula C6H12O6, 6 carbons, 12 hydrogens, and 6 oxygens. So we'll just slowly draw this molecule for you. So in this first section of the diagram here, I have it as a linear molecule, just to help our understanding. And the first thing to remember is that each carbon is going to be joined together. These are covalent bonds. And of course, we should recall that the valency of carbon is 4, that of hydrogen is 1, and our oxygen valency is 2. So we need to incorporate that into our thinking. So in order to draw the structural formula of glucose, we are going to connect carbon 1 to carbon 5 via an oxygen. And we can see that structure here. So here's carbon 1, and here's carbon 5, and there's the oxygen used in order to join them both together. And so now we have a ring structure to glucose. glucose takes very many forms, some of them linear, some of them ring structures. And so we're just looking at a few of the common ones which occur in biology. So this ring structure is glucose. But we still have to account for, although we have our 6 carbons and we've taken care of 1 oxygen, we still have the hydrogens and the oxygens to fill in. And the routine is that for each carbon, so here's carbon 1, we add a hydrogen and an OH group. And we're going to do that to all of the carbons. So let me just do that. Hydrogen, OH. I should pause and just tell you that when I say OH, what I really mean is oxygen bonded to hydrogen. So there's the valency 1 of hydrogen, like that. This carbon, it actually reverses, so we're going to get this, and then back to the original OH group down. So notice that the OH group is down, down, up, and then down. And that's because what we are actually doing here is we are drawing a form of glucose called alpha glucose. Usually we'd use this symbol, alpha glucose. However, we're not finished. We have to add an H group here and an OH group here. Now if we then count up the hydrogens and oxygens, we'll see that let's do the oxygens. So we've 1, 2, 3, 4, 5, and 6. So remember it's C6H12O6. So we've got all our carbons, we have all our oxygens. But if we look at this 5th carbon, we still need to add a hydrogen here. And on the 6th carbon, we also fill in with a hydrogen. And that would take care of the 12 hydrogens required if you go through the diagram and count them. So connect 1 to 5 to make the ring. Then add H and OH, hydrogen and hydroxyl to each of the carbons, and then fill in with hydrogens to complete the valency of 4 for carbon, 1 for hydrogen, and 2 for oxygen. As you can see, the drawing of the diagram starts to get rather complex. This is pretty much what we just drew, which is the alpha form of glucose. But in textbooks and in your reading, you may very well start to come across simplified forms. You can see here that the carbons are indicated by the numbers at the vertices, and the orientation of the OH groups are shown by filling those in. And there's a further reduction and simplification in this structure here. For the purposes of an examination, you would probably be drawing at this end, but perhaps in an essay where you're dealing with polymers, you might be drawing at this end. I've emphasized the fact that the first carbon in alpha glucose, the OH group is down. Contrast that with what's called the beta form of glucose, in which on the first carbon, the OH group is up. So it's up here, up there, and up there relative to the rest of the molecule. So for comparison then, we have down, down, up, down for our OH groups on the first 4 carbons, where here we go up, down, up, down. So it's actually only the first carbon where there's a difference. So alpha and beta glucose are known as anisomers. They have the same formula like this, but their structural arrangement is slightly different out here on carbon 1. Glucose will come across in anabolic processes where it's made in photosynthesis, particularly in the Calvin cycle. And we'll also be seeing glucose again when we break it down in the catabolic process of respiration. This is a relatively sweet sugar. It's very soluble in water, gives you a positive Benedict's test in which the color change in that test is from blue to orange. Being soluble, glucose creates osmotic issues, and it's also known as a reducing sugar, and perhaps we could use the phrase biologically reactive. It tends to get involved in chemical reactions. More later. So below you'll see the appearance of links to additional videos in this sequence, the possibility to subscribe, and links to the playlists and channels so that you can look for the videos that you need to support your studies.